কেমন আছিস? Ishendu, please go on. Is it audible?
Yeah,
Ishendu, go on.
okay. Good morning everyone এবং রৌদ্র করজ্জ্বল এই সকালে সুপ্রভাত। On behalf of Department of Physics, Bhairab Ganguly College, I, the convener of International Webinar on Biophysics, welcome you all. We have a speaker from University of California, Dr. Ronojoy Saha. He will deliver a talk on the emergence of RNA world. So I would request Dr. Deobrat Bhadro to introduce Dr. Ronojoy Saha.
Hello and good morning, all of you. It's indeed a privilege to welcome Dr. Saha, who is my close friend, or rather childhood friend. So, a little bit about biophysics. Biophysics is something which is much more unexplored now, and the work going on is very much in the rudimentary level. So, what biophysics has been critical in understanding the mechanics of how the molecules of life are made, or how different parts of a cell move and function, how complex systems in our bodies, the brain, circulation, immune system, and others work. So, biophysics is a vibrant scientific field where scientists from many fields, including mathematics, chemistry, physics, engineering, pharmacology, and material science, use their skills to explore and develop new tools for understanding how biology or life works. So it's a kind of the bridging science. Yesterday I was looking for some literatures where the different fields, so what not, like data analysis and structure, computer modeling, molecules in motion, neuroscience, and biomedical applications, bioengineering, nanotechnologies, imaging, ecosystem, what not are being assimilated in the subject. So in this topic, I am not the expert. The expert is today's invited person, who is from University of California, Los Angeles. So let me introduce Dr. Saha. Dr. Saha did his graduation in chemistry, then he shifted to biotechnology and did his degree from Jadavpur University, followed by his PhD at SN Bose Center for Basic Sciences, Kolkata, in biochemistry in the year 2014. Then he moved to US. He is now the assistant project scientist at the Department of Chemical and Biomolecular Engineering, University of California, Los Angeles. Previously, he were associated with the University of California, Santa Barbara, where he used to do his research work with RNA activity inside vesicle protocell or UV radiation on RNA aptamers. In his doctoral work, he has the experience of structure and function of protein in cell-like confinement and dynamics of confined water. So he has a long career of research, doing numerous publications in high-impact journals, most reputed one, and the H index of 10. So with this brief introduction, I would like to request Dr. Shaha to deliver his lecture. Please, Dr. Shaha.
Thank you, Deobrata, for the nice introduction, and I'm also I'm very much glad that that the organizing committee has given me this opportunity to speak on my research on RNA world. Okay, so without any further delay, I would like to start. Let me share the. Okay, is my slide visible?
Yes,
I
perfect.
hope my slides are visible.
Perfect.
Yeah. Okay. Just a second. Okay. So my talk today basically consists of sorry, just a second. Okay. Today's talk basically consists of two parts. First, I would like to introduce you about RNA world and the origin of life. So RNA world, I mean, basically it's a there is a very good collection of data that suggests that um, during 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 the prebiotic evolution, there was only RNA molecule. Like biology came from RNA, and then basically gradually protein and various DNA world gradually evolved. Moving on, in the second part of my today's talk, I'd like to give you like some basic idea about the potential role of environments on the emergence of this RNA world. I would like to talk about some of our experimental findings, like how environment could actually could influence the emergence of this RNA world. Okay, so before I start, a very I think I need to introduce you what is RNA. So it's basically RNA is almost identical to DNA, I would say. So we all know about DNA, right? DNA is our hereditary molecule. It carries genetic information that helps us help us make protein, and protein is the cell's workhorse. Like carries out all the biochemical function in our body. Okay, so DNA actually carries that information and passes this information to protein. So we all know these things, but if you look the structure of DNA, it's basically a double helical structure. It's a polymer of nucleotide molecules. In other words, we say it's a polynucleotide composed of two polynucleotide chains, which are basically joined by hydrogen bonds. It's a double helical in nature. RNA is very much similar to that, but in comparison to DNA, which is double helix, RNA is a it's composed of a single nucleotide molecule, single polymer, composed of a single polymer. Besides that, another difference between DNA and RNA is the difference in the bases. So both DNA and RNA consist of the bases, as you can see here. So, but all most of the bases are similar between DNA and RNA, but there is one base which is called uracil, which is actually different in DNA, and it's called thymine. So besides this, there is another important difference between DNA and RNA, which you can see here at the right side. Like if you look at the chain, in RNA, there is a OH group present, and extra oxygen present at the five-membered sugar ring, which is absent in DNA. So, and that's why it is called DNA is called deoxyribonucleic acid, whereas RNA is called ribonucleic acid. And you'll see like this presence or absence of this oxygen has important consequences in biology. So because if people have found that, like because of this extra oxygen at this position, RNA is much more susceptible to degradation. So RNA is it's found that RNA is million times less stable compared to DNA. So DNA is much more stable, and that is the reason we see like DNA carries our genetic information. So the idea is if you want to pass information from one generation to next generation, your information carrier needs to be stable, and we see like DNA is actually much more stable compared to the compared to RNA. Okay, so what happens? We see like DNA carries our genetic information. It passes information to RNA through transcription, and finally RNA transfers that information to ribosome, um, and ribosome make protein. Okay, so this is called the central dogma of molecular biology. But there is a puzzle here. This is actually very complicated. If you look, this whole process, the central dogma is very much complicated, and each step depends on the other step. So you consider the replication of DNA. That means copying of DNA. That is important for making during cellular division. Like you need to copy your DNA, you need to multiply it, and this replication process is dependent on protein. Okay. Similarly, next you consider the transcription of DNA that is involved to make to synthesize RNA. That also requires some protein molecule. And similarly, the final step, the translation step, which occurs on ribosome, it also is dependent on protein molecule, because ribosome, you'll see, it's basically nothing but a complex of RNA and protein. So this puzzle I would rather say it's a paradox. In the context of origin of life, people were very much like puzzled, like it is quite puzzling to see how this complex system could evolve. This is kind of a chicken and egg problem, like which came first? Is it the DNA or the protein? Because each step is dependent on the other. In order to do each step, you need protein. So this is a chicken and egg problem. So people were very much were quite puzzled how this complex chemistry could actually evolve during origin of life, like in the prebiotic origin of life. But now the answer to this complicated question was actually solved later on in 1980s. Two famous biologists, Sid Altman and Tom Cech, they had a very interesting finding. Like they independently found like not only like protein, like they found like RNAs can act also like as protein enzymes. So they found during that understanding was only protein can act as an enzyme, but these two smart people, they found like RNAs can also act like an enzyme. Okay, so Sid Altman, he saw like there is some RNA molecule which is called RNSP. So what it does, it can go and it can bind some certain RNA molecule like tRNA molecule, which is shown here in the red color at the bottom. So what it does, it binds to this RNA and it cuts it. It cuts the tRNA molecule, and this cutting event is if they found like it's billion times faster when the RNSP molecule is present. Without RNSP, this cutting event is much slower, but in presence of RNA, like RNSP, it is much faster. So this is an example. So we can say like it's where RNA can act as an enzyme molecule. Similar, another example actually came from Tom Cech. Tom Cech at University of Colorado Boulder, he found like mRNA maturation. So what is mRNA maturation? So it's, I would say, like in our body cell, there are lots of junk or DNA sequences, which is not functional at all. It doesn't code any protein molecule. So what happens during the protein synthesis is actually this mRNA molecule like cuts out those junk RNA. Sorry. Okay, so cuts out those junk RNA, and these junk RNAs are called intron. And then what happened? Like these, you can see here, like these two other RNA molecule then joins, and this is kind of a trans-esterification reaction. And Tomczyk observed that this trans-esterification reaction can be self-catalyzed. So it can be self-catalyzed, and so self-catalyzed. And so this is another example of RNA molecule where it is functional. So these two example is well known in literature, and it's actually was very significant with respect to origin of life and as well as RNA world. And actually, because of this discovery, these two guys got Nobel Prize in the year 1989. Okay. Okay, there are other examples where people have shown RNA can act as an enzyme molecule. If you look the protein synthesis in biological cell, you'll see it happens in ribosome, and the ribosome molecule is nothing but a complex of RNA and protein. And if you look at the core of this ribosome molecule, where this protein synthesis actually take place, it's basically nothing but RNA molecule. So the red region here, to zoom it out, and you can see this is nothing but RNA sequences. So you'll see one of the very important reaction in biology, like protein synthesis, which like is our cells workhorse. It's actually synthesized by an RNA molecule. Okay, so not only that, there are other example of RNA being the enzyme molecule, like in bacterial cell. There are certain RNA molecules which are called riboswitches. What is riboswitches? Riboswitches is something like is nothing but some regulatory element. Why regulatory element? Because it's present at the pipeline mRNA region of most bacteria, some bacterial species. And what it does, it binds some particular metabolite, like say we call ligand, and the conformation of this binding region changes upon ligand binding, and this changing in conformation has important consequences on the expression platform. Basically, what happens upon metabolite binding or ligand binding, the structure of this region, the regulatory region changes, and that's why protein expression changes. Okay. And people have found like there are multiple such riboswitches present in bacteria, and these are actually very much significant with respect to with respect to the physiology of bacterial cells. Like there are certain riboswitches like thymine riboswitches, which can actually help in the biosynthesis of vitamin B1. Similarly, we have example of flavin mononucleotides, which actually regulates vitamin B2 biosynthesis. So there are numerous other examples where riboswitches could actually influence the gene expression of a bacterial cell, and that has important significance. So far, I think I have at least I believe I have been able to convince you like RNA can act as an enzyme molecule like proteins. Okay, so moving forward, so now our research question is, how did this functional RNA evolve? So what are the conditions, like at what condition like this RNA could evolve? So if you look the early history of life, like that, I mean the timeline of events that led to the current biological cell, you'll see we start from let's say we start from formation of Earth around 4.5 billion years ago. Earth formed, then gradually Earth cooled down, and we started forming we started getting stable hydrosphere because water condensed and water layer form on our Earth, and following that, at some point we have prebiotic chemistry. due to some certain environmental condition, we think that led to all the molecular building blocks of life, like say amino acid or say RNA or something like that. I mean, not RNA, I mean the nucleotides and all. So all the molecular building blocks of life actually form around this age. Then eventually, like we think like pre-RNA world, like RNA-like molecules actually formed by certain way, and then this formation of this RNA wall then led to the formation of functional RNA wall that we currently see, and then at some point, like cellular life evolved around 3.6 billion years, 3.6 billion years ago. Okay, now our question is like we think like there is, as I mentioned, we think there is a pre-RNA world, and we think and there is also a complex RNA world that we currently encounter. Now our question is how this complex pre-RNA world actually evolved to form the more complex RNA world. Okay, there could be multiple factors. I think environment. We think environment might have played a very dominant role, because we all know like environment during prebiotic condition was totally different. It was much more harsh condition, temperature, pressure, pH, everything was totally different. So in the next few slides, I would like to show you some of the possible, I would say plausible environmental condition like during which life might have evolved. Life, I mean in other words, RNA world might have evolved. Okay, so moving on. So this is an example. So this is iconic picture, an iconic picture of deep sea hydrothermal vent. So this is a condition like where temperature and pressure are extremely high, but you will see like surrounding area is full of life, like various kind of microbial and also aquatic life is there. So this is a classic example where life is sustaining at various high temperature and pressure. Okay. Another example is this is the picture of Rio Tinto River in Spain. Like this is a very acidic condition. Environment is very much acidic. pH is very low, around 1.7 to 2.5. And here also you can see like life can sustain. Different kind of life is there. So this is a very classic example of life at acidic pH. There are other examples of life, like life at high temperature as well. This is the image of this is the hot spring of Yellowstone National Park, and here also people have you can see there it is full of life, both various kind of microbial and also aquatic. So so we think like this kind of condition could actually like since at those conditions current life is sustaining, we think like those conditions also could actually modify the evolution of RNA. Okay, another plausible condition we think is UV. So we know like UV flux at Earth's surface was like around 3.9 billion years ago was 2 to 3 times order higher. And I think but a common misconception regarding UV is like we all know like solar UV light is lethally bad for life's building blocks, like as well as life, like it damages DNA, we know. But there are some other example, but with respect to origin of life, people have actually shown like UV light is actually sometimes helpful, but because they have found like UV light could actually help synthesize the molecular building blocks of life. So you take example, so you start with hydrogen cyanide, you shine UV light, okay, you shine UV light on it, and what you end up getting is very interesting. You can, through organic reaction, you can actually form sugars, which is important building block for carbohydrates. You can also synthesize pyrimidine ribonucleotides. It's all, of course, one of the molecular building block of RNA. Then you can also form lipid precursor. Lipid is important for our biological cell, because all our biological cell is composed of phospholipids. So lipids can be prepared, can be synthesized from hydrogen cyanide in presence of UV light. Not only that, our building blocks, like the building blocks of proteins, which is very important for our biology, for our human body, is actually can also be synthesized. Like through this synthesis pathway, actually they are able to synthesize 10 different amino acids. So this is a classic example where like UV light was actually helpful for prebiotic synthesis of molecular building blocks. So motivated by this work, we were actually interested to see how how UV light could actually modify the structure and function, and as well as function and replicability of RNA enzyme molecules. Okay, so moving on. So we plan to choose some functional RNA molecule that we call RNA aptamers. What it does, these are not like natural RNA enzymes, but these are artificially selected RNA molecules that are chosen to bind a particular target molecule. So in this case, this target molecule is so in this case, the target molecule is some fluorescent molecule. But what is interesting is that this fluorescent molecule it doesn't have much fluorescence in free condition. So if you take the spectra of this fluorescent molecule in buffer condition, standard buffer condition, you'll see the fluorescence is very low. But what is interesting is like when this fluorescent molecule bind its particular aptamer, like the selected aptamer, I would say, the fluorescence of this dye molecule increases significantly. So there are various such examples of RNA aptamers, fluorescent RNA aptamers. We plan to choose these two RNA aptamers. One is called malachite green that binds the dimalachite green, and there is another aptamer which we call spinach aptamer that binds this particular dye DPHBI1T. And in both the cases, you see like binding increases the fluorescence. So monitoring the fluorescence, so we can say like we can actually study its activity, the activity of this RNA molecule, which is functional. Okay, so what interesting we found, like we've actually monitored the fluorescence of this RNA aptamer molecule with changing with the time of UV radiation. So we exposed these two aptamer in presence of UV, and we monitored, kept monitoring the fluorescence, which is the signature of its functionality. And what we found interesting is that like both this aptamer molecule actually loses its fluorescence. Okay, in both the condition you'll see as the time passes, the fluorescence goes down. But what is interesting here is that we found like the rate of fluorescence decrease is different. For malachite green, which is blue, is much faster compared to spinach, the other aptamer. And what is interesting is that we found like at some point, like at 6 hours, we got like almost like all the fluorescence of this malachite green aptamer vanishes. It's totally destroyed by a UV exposure. But however, for spinach, we saw like there is still certain fluorescence is still there. It's not totally zero. Okay, so this was a very important, interesting observation to us. It looks like different RNA molecule is behaving differently with respect to UV damage. So we wanted to study this in the molecular level. We wanted to see what's going on, what's the structure, how the structure is impacted by UV radiation. We monitor the thermal stability. Thermal stability of these two aptamer, and we found interestingly the stability which dictates functionality of this RNA aptamer molecule is actually modified. We found melting stability is much higher before UV radiation, but after UV radiation there is a significant decrease in melting stability, as you can see. And we confirm this using polyacrylamide gel electrophoresis. And what we found interesting is that, like due to UV radiation, we saw another band, extra band in the gel, which suggested like due to radiation, the structure is disrupted. Okay, the tertiary structure of the RNA is disrupted. Okay, so we also studied the same stability of the other aptamer, as I mentioned, which was retaining its activity. But here, we very interestingly, we found like this aptamer, the stability, it doesn't change due to UV radiation. Okay, so I think this is consistent with the functional damage that we are seeing due to radiation. Okay. So moving on, we wanted to study what's going on, like what is happening, how like basically wanted to see where the damage is happening, like what kind of damage is going on. Okay, so we actually used RNA copying chemistry, like reverse transcription, which is a well-known technique now to detect COVID-19 infection. Or basically, using the same idea, we actually monitored the modification, basically on RNA copying chemistry. So you see here, like if there is modification in the bases, like your RNA copying will be stalled, and that can be actually imaged by running a gel, by running polyacrylamide gel. And very interestingly, what we found is like UV damage is very much higher. So here, like the area of the circle is proportional to the amount of damage that we encountered. And so by this technique, like reverse transcription technique, we were actually able to see where the damage is happening and how much is the extent of damage. And interestingly, what we found is that the damage is mostly in the binding pocket. So what do you mean by binding pocket? Binding pocket means where the dye is binding the RNA molecule. So this is the secondary structure of this RNA aptamer, the malachite aptamer. And what we found is that like damage is more prone in the binding pocket compared to where in the stem region. Stem region means what? Like which are much more folded region, where which are hydrogen bonded region. So this is the stem. So this is the stem region. These are the stem region. So you see, the damage is much less here, but damage is more in the binding pocket. Okay, so this was interesting to us. We also like analyze this gel data in order to see how the reactivity of this bases changes along the RNA molecule, and we found consistent result here as well. Like we found that damage is much more prone in the binding region compared to other region. Okay, so long story short here. So in conclusion of this experiment, we found like UV exposure creates. We found that UV creates chemical lesion in RNA, because we found like RNA copying is stopped. So there is definitely some chemical lesion formation. That's why UV like copying is stopped. But we found this is it's a sequence dependent phenomena, because we found we actually monitored two different RNA aptamers, and in one case we found much more damage. The rate of damage is higher, as well as the extent of damage is much higher. However, in the other case, the rate of damage is much less. And we think like there could be multiple reasons could be possible. One could be structural. Structure of these two aptamer could be different, and lesion formation could be mutually explosive, or there could be some self-repair mechanism going on, and that's why one is sustaining its activity, whereas other is much more prone to damage. Okay. Another conclusion, I would say, like we saw, like copying chemistry is actually like hindered by UV damage. So we can say like templating ability of RNA, which is one of the important characteristics of the RNA world hypothesis, is actually is influenced by UV damage. And finally, we found like susceptibility of damage is much more in the binding pocket, like where the RNA is binding the dye, compared to the stem regions, like where it is much more hydrogen bonded. So we can say like during evolution, the evolution of RNA, well-folded RNA was much more favored compared to those unstructured region. So unstructured RNA were much more favored during origin of life because they were able to sustain its activity. In other words, it was less prone to damage compared to less folded structures. Okay, so the final take-home message of this experiment is like UV exposure could actually act as an important selective pressure. We think due to origin of life. Okay, moving forward. So, and in another study, we wanted to see see how other environmental conditions could actually affect the evolution of RNA. So we thought like biological cell would be an obvious such environmental condition, because if you should see modern cell, it's basically like all the biomolecules like RNA or protein, everything, DNA, everything is confined within a lipid enclosure, right? And this is called so it's like composed of phospholipid molecule, and like our lipid bilayer is composed of phospholipid molecule, and it's kind of a confined condition. So we wanted to see how this confinement condition was actually could modify the activity of RNA molecule. We think like similar condition, like cell-like condition, was actually present during origin of life, and this where like you can see like a lipid enclosure is containing RNA molecule, and this is called protocell. Means it's kind of like it's not as complicated as modern cell, so that's why it's called a protocell. So the idea is a confined condition, and how this confined condition could modify the evolution of RNA. Okay, the first obvious question is whether these protocells were actually possible in the context of origin of life. Okay, that is a very obvious question. Whether can we actually synthesize protocell in prebiotic condition? The answer is yes. So people have actually, these smart people like Professor Dimmer actually were able to synthesize, able to extract some certain lipids like various fatty acid and phospholipids from those various meteorites, mostly fatty acid. And when those fatty acids were actually were added to water, they were able to synthesize. They're able to actually look vesicle-like structure. Vesicle means like cell-like structure, which we call protocell. So they found like protocell-like structures are actually possible in prebiotic condition. Like from meteorites, you can actually synthesize. You can actually prepare those cell-like structures. So this was interesting, and other people like Professor Sostok and group actually shown that like these protocells can actually undergo growth and division. So interestingly, if you suppose you have some vesicle or protocells, you add lipid to this. At certain condition, it forms this overgrowth, and you move on, and it forms these lamellar-like structures. And then if you give some mechanical stress to it, or if you give some mechanical strength, like you shake it, it can actually divide to form various other protocells, okay, or called vesicles. So this is a classic example, like where like people think like in prebiotic condition, those protocells can actually not only undergo growth, but they can also undergo division, and this is a prerequisite for biological cell division. Okay. So as I mentioned, so we wanted to study how this cell-like condition could actually modify the evolution of RNA. So we actually encapsulated certain aptamer molecule like malachite aptamer inside vesicle-like structure formed from fatty acid molecule, and we wanted to see how this activity is different when it is present outside the vesicle. So with this question in mind, we chose malachite aptamer. I think I already introduced you with this aptamer. So moving on, we actually monitored the activity of this RNA aptamer molecule in two conditions. One is inside vesicle, and another is outside vesicle. So what we found interesting is like when the RNA aptamer molecule is present inside vesicle, the binding activity is much higher. So it is the if you look the binding curve, it's much more shifted to the left. So that indicates like activity of the RNA molecule inside this fatty acid vesicle is much more higher. Okay, so we tried this. So we actually tried other confinement condition, like we tried other vesicle. So in order to see whether it is something related to the interaction, like chemical interaction of the RNA with the lipid, or it is like more general phenomena. So we wanted to study that, and we changed our membrane composition. We used different other fatty acid, and in all the conditions we found like inside vesicle, the binding activity is the functional activity of this RNA molecule is much higher, at least fourfold higher. Okay, so this is some other example like where we employed lipids, like lipids, I mean like phospholipids that our cell is kindly made of. And in those conditions also we found like activity is much higher. Okay, so far so our understanding is like activity or functionality of RNA molecule can be increased inside vesicle or inside protocells, but we wanted to study what's going on, what is the how the structure is actually affected due to presence of those RNA molecule inside vesicle. So we monitored the stability, melting stability or thermal stability of the aptamer inside vesicle and compared it with respect to when it is present outside. And very interestingly, we found like encapsulation increases the stability of this functional RNA molecule. We found like it is much more compact, like that in like when it is RNA aptamer is present inside vesicle. So we wanted to study this in more detail. Like wanted to see where like which bases are basically modified due to encapsulation. We employed RNA copying chemistry, like which is called shape. I'm not in short. I'm not going in very detail. In short, the idea is you basically you need to modify each basis of the RNA with some acylating agent, but the acylation will be affected depending on the flexibility of the basis. Whatever is much more flexible will be acylated more compared to the other one which is less flexible. So with this technique and using RNA copying chemistry like reverse transcription PCR, we actually were able to pinpoint the stability of the I would say the flexibility of the bases inside and outside vesicle. And what we found interesting is like some of the bases is actually modified due to encapsulation, and and this and monitoring the flexibility of the bases with respect to the concentration of the ligand or concentration of the ligand molecule that it binds. We found like inside vesicle, like the molecule, the RNA molecule is much more. The structure of the RNA molecule is much more compact. So it's much more compact means like it's much more functional, and it's much more compact means much more functional. So so we did some theoretical calculation basing we considered this RNA as a freely like polymer which are composed of freely jointed chains. We did some simple calculation and we found like our calculation is very much consistent with the observed value. With the, our observed value is very much consistent with the predicted value. So basically, the idea is the theoretical idea is RNA molecule. It can be like when you have RNA molecule in solution, it has two possible conformation. Okay, one conformation is like it could have a very compact structure, and it could also have a very extended structure. Okay, and now when you employ like cell-like confinement to this ensemble of structure, so since cell has a limited volume, so this compact structure of RNA will be much more favored compared to the extended structure. Okay, because cell has some certain limitation. It has volume restriction, and this is called excluded volume effect. So due to excluded volume effect, we think like the folded structure or the compact structure of the RNA will be much more favored, and that's why like the like we see like different functionality of the RNA, because conformation most favored, like more compact structure are much more favored inside vesicle, and that's why you see much more functional RNA. Okay. So to put this in another word, we see like we think like vesicle could act as a chaperone for RNA. So we all know like I think most of some of us know like protein could act as a chaperone. So in biological cell, proteins can be misfolded. It can be non-functional, but there are certain protein molecules that are actually help make this protein like help make this protein functional. So this favors the misfolded structure of protein, and these proteins are called chaperone. So the proteins which actually folds the other proteins, this is called chaperone. We think like vesicle or protocell condition could act as a chaperone for RNA to confinement effect. So what happens? What it does? It disfavored the unfolded structure, but without affecting the folded structure. So the delta G of the folded structure is not affected, but unfolded structure is disfavored due to confinement effect, and this destabilization of the unfolded effect gives relative stabilization of the folded state. So that's why we get more functional, more folded RNA molecule inside vesicle, and that's why we get more functional RNA molecule. So in short, we can say vesicle encapsulation was beneficial for the evolution of RNA enzymes. It, as I mentioned, is favors the folded structure, and that's why it makes more functional RNA molecule. So summary of today's presentation. So I think I have been able to partially convince you like biology might have come from RNA. So at some point RNA was the sole biochemical, sole biomolecule which carried out not only, which basically not only acted as an information carrier like DNA, but it was also able to carry some certain enzymatic function, which was important for cellular evolution as well. So for this, I think I presented you some example, like classic example of ribozymes. I mean RNA enzymes like RNSP, intron, then various ribosomes, and then riboswitches, which showed us like RNA could actually act as an enzyme molecule. Okay. Next, I think I actually showed you some of our efforts in order to understand like how prebiotic environment could actually modify the emergence of RNA world. Towards this, we found like protocell encapsulation was actually beneficial for the RNA evolution, and also we found like UV radiation, depending on the sequence of RNA, it could also take a important role in evolution of RNA. So with so I'd like to stop here. So this is the overall picture of the campus, or like University of California Santa Barbara campus, where I did my research. It's nicely located by the by Pacific. So, and this is the famous Henry Gate, and this is the like aerial view of the campus. This is our department, chemical and biomedical engineering department, where our lab was there. This is the current. This is the this is our lab like group picture, and you can see me in action. Okay. With this, I'd like to acknowledge the whole chain lab and specifically Professor A. Chen, who gave me this opportunity to work on this fascinating field of origin of life. And of course, I'd like to thank the founding agencies, Simon's Foundation, Institute of Collaborative Biologies, as well as NASA Astrobiology for funding, and also UCSB and UCLA, where I'm kindly working on. Okay, I'll stop here, and I'll be happy to answer any question if you have.
Thank you, Dr. Ronojoy Shah, for giving nice presentation of your research. অ্যাকচুয়ালি বাইরে প্রচন্ড বৃষ্টি হচ্ছে এবং তার সাথে আমাদের চ্যাট বক্সেও কোশ্চেনের বৃষ্টি হয়ে গেছে। তো আমি একটা একটা করে কোশ্চেন করছি, প্লিজ অ্যান্সার দেবেন। তো প্রথমেই একটা শর্ট কোশ্চেন এসছে ফ্রম আমাদের দেবব্রত ভদ্রর কাছ থেকে, যে হোয়াট ইজ জিআরএনএ? ক্যান উই ডিজাইন এ? ওকে, ঠিক আছে, হোয়াট ইজ জিআরএনএ?
Okay, I'm not. I think it's some kind of a modified. This kind of RNA that is made of modified nucleotide. Not exactly sure what is that. I've heard of it, but I don't know exactly what it is. But it is definitely some polynucleotide, ribonucleotide molecule composed of some some different modified bases being present.
Okay. Can we design a DNA spinner aptamer instead of RNA?
Yeah, that is possible. Yeah, I don't see any problem in making that. Yeah, basically you have to go through artificial selection strategy, like that. Yeah, there are example of DNA aptamers. So people have been able to synthesize like select DNA aptamers which can bind a particular target. So I don't see any problem in making that.
Okay. Another question from Monisha Mondol. Sir, what does RNA world hypothesis suggest?
So yeah, so RNA world hypothesis. I mean, so currently what we see, like if you see central dogma, you see information transfer starts from DNA to RNA, then RNA makes protein. But in the context of prebiotic evolution, we think like this complicated sequence of reaction was not possible because this is a chicken and egg kind of thing. Like if you do not have protein, how will you make? How will you do this whole thing, like from DNA to RNA and RNA to make protein? Everything requires protein, but if you do not have protein, how will you make it? So the answer came from the discovery of ribozyme or RNA molecule, RNA enzymes. So this like people suggested like RNA, it has capacity, like it can carry genetic information like DNA because it has base sequence. Of course, there is stability issue. That's why in today's world, like we see DNA being the information carrier. But at that point, we think like DNA was the information carrier. But but by the time, meanwhile, it was also able to simultaneously. It was also able to act as an enzyme molecule, which is important for biochemical reactions. So this is RNA hypothesis. Like people think like everything's biology started from RNA because it has both these two things. Like it can carry information, it can act as a like it can self copy itself, it can catalyze biochemical reaction. So everything started from RNA, and then DNA and protein evolved.
Another question from same person, that can a ribosome self-replicate?
Yeah, so people are actively working on this field. They have been able, so far my knowledge, they have been able to copy RNA, maybe some short RNA molecule, say 10-15 bases, but people are still continuing to yes trying so to extend this like to make the long RNA molecules. So as Debrot was asking like tRNA, so people are also trying various modified nucleotide molecules, which and trying to and get actually investigating whether the modification of the bases RNA bases could actually increase the self-replication ability, like self-copying ability of RNA or ribosome. Yeah, very good question. Yeah. People are still trying.
Shayoni Dotto asking that UV rays are harmful for eyes. We have studied. Mm-hmm. So, as a whole, are UV rays good for human? This is a general question.
Yeah. So basically, as I showed, like definitely UV is bad for DNA damage, and also I showed you like from our study we found like RNA is also damaged by UV. But what we found interesting is like rate of damage could be different depending on the sequence of the RNA being impacted. So that is one thing. The idea was, as I showed you, like UV was in some sense UV was good for making the molecular building blocks. Okay, like you could see, like you can synthesize all the molecular building blocks, amino acid, ribonucleotides, then like lipids, everything. So in one sense, maybe in the context of RNA, context of origin of life, it was helpful, but it is certainly AV is bad. AV is detrimental for cell, biological cell, at least in today's cell, like modern cell.
Another question from Olivia Mukherjee that what are the basic theories used in confinement?
Basic theory, I mean, so it's like, so as I said, like confinement means if you have a restricted volume. So this is famously called excluded volume effect. So what it does, as I said, like both RNA, like RNA as well as protein, like they have always like if you look those biomolecules in standard condition, like buffer condition, you see there is an equilibrium between a folded state and unfolded state. Folded state means it's much more compact, and unfolded state means it's much more extended. Now, what is the current idea is like um, when you encapsulate this RNA molecule or protein molecule inside those confined condition, the unfolded structure will be much more will be much less favored because you have a restricted volume. How come extended structure will survive there? So idea is, so what will happen, like the whole equilibrium will shift towards folded state, right? So that is the idea, like due to confinement condition or excluded volume effect, folded structure will be much more favored, and that's why in our study I think we found like uh, RNA functionality is much more elevated within such excluded volume condition or physical condition. Yeah.
From Shubhankar Banerjee, that when malachite green is inside vessel, how it increases activities of RNA and how we are detecting it by fluorescence.
Okay, so basically what we did is like we encapsulated the RNA molecule inside vesicle. We have a way like size exclusion chromatography we actually use to purify to sort out only vesicle encapsulating RNA molecule. And what we did, and then we added the dye molecule, the malachite green dye from outside, and allowed it to incubate overnight. So it will diffuse to the membrane. We checked it like it can diffuse to the membrane, and basically at the end it binds those aptamer molecule like malachite in aptamer. And then what we did, we monitored we collected those vesicle, and we monitored in a cuvette, we monitored the fluorescence. Actually not cuvette, we actually used some plate reader to change the concentration of the dye and monitor how fluorescence is changed inside vesicle as well as outside vesicle. And after plotting the fluorescence with respect to concentration of the dye, we were actually able to get the binding constant or the KD vector. And the idea is higher is the bind KD, lower is the activity.
Three questions are from Joydeep Chatterjee. That does RNA have solar energy retention and charge transfer characteristics or magnetic hysteresis, which can be suitable for its application in the field of dye-synthesized solar cell or battery.
Okay, can you please repeat?
Does RNA have solar energy retention and charge transfer characteristics or magnetic hysteresis, which can be suitable for its application in the field of the dye-synthesized solar cell or battery?
Yeah, it's an interesting question. Actually, I do not have much idea about it, but definitely, like RNA can absorb UV radiation, so it might have some impact. But whether it will be it can act as a dissociative solar cell, I'm not aware.
Okay. Another question is, how does the spectral characteristics of RNA change with the variation of the solvent or buffer solution?
Spectral characteristics of RNA.
Change with the variation of the solvent or buffer solution.
Yeah, so RNA being a polar molecule, it will definitely the spectral properties like I mean maybe ask like question is on UV like on the absorbance spectra. I believe. So yeah, I think it should change, but how much? We didn't see much change when we were like in our condition. The changes was not that much, but definitely like changing polarity hugely might impact it.
What are the features should we look for RNA fingerprinting?
Uh, I do not have much very good answer for this.
So one general question from Sujay Saha that I am in physics field. Is it any scope for me in biophysics research area?
Yeah, obviously, like it depends like what kind of like there are multiple uh, like research option. Like, it depends like what kind of research you want to do. Like maybe if you want to like biophysics is a huge field, so yeah, there are definitely possibilities.
And, and the last question is, can we design aptamers that target other nucleic acids?
Uh, yeah, I think it is because yeah, I know like people have used aptamers which can target proteins. Uh, then why not? Yeah, I think it's possible. Yeah.
So we don't have any question now. So we have so thank you, Dr. Ronojoy Saha, for giving a nice research, and the question answer round was quite informative. So once again, thank you very much. And for dear participants, we have sent feedback form already. Please fill up, and certificates of this webinar will be issued within 4 days. Thank you very much. Thank you all of you.
Thank you, everyone.
Thank you, Ranojay.